इतना बाद में खाते हैं। So, good evening, everybody. Today is the last class of course completion. Very good evening, Justina. Yeah. So, today is the last class of course completion. Good evening, Ajay. We will also have one more class. So, next week we'll have one more class. I think it will be on Tuesday. Most probably, your next class for the next week would be on Tuesday, 7 to 9 p.m. Exact notification would be sent to you through your mail IDs. So, today we'll be continuing the discussion that we did in the last class. And so, we were studying about little bit about the evolutionary aspect of developmental biology, how the evolution of various organs or the various kinds of forms have taken place. So, when we study about the evolution, small genetic changes are acquired over the period of time. Okay, that we all know that small genetic changes are acquired over the period of time, but these small genetic changes are many a times not sufficient enough for the generation of some novel structures. Okay, so if we want, or if the nature wants the generation of certain novel structures, then just mere the accumulation of small genetic changes is not sufficient enough. So that means there has to be some other changes as well that can accompany with the small genetic changes that happens over the period of time, so that some novel structure, evolutionary novel structures can be formed, and particular organism can adapt itself according to its environment. So the key differences are not in the alleles of protein coding genes, but in the sequence that decide where and how much the genes are activated. It is important. What does it mean? It means that, see, just a minute, I'm searching for my pen. Yeah. So, if we start with the, I'm not going to give a very detailed introduction just before starting the questions, it's just like a basic. Okay. Okay. Hmm. So we all know that first is your DNA, from that forms your RNA, and then it forms your protein, correct? So that means that whatever phenotypic changes that we see are actually because of the expression of the proteins that has taken place, or the formation of the translation of the proteins that has taken place. But you have to understand that protein will code according to the information that has been given by its conjugate or cognate RNA, and RNA will be transcribed on the basis of whatever the sequence of DNA is there. So the baseline is that changes in the sequence of DNA is going to reflect ultimately what kind of protein is formed. This is very important. So that is why whenever we study about mutations, we study about mutations that happens mostly, not all the mutations, but we mostly study about the mutations that happens at the DNA level that can affect up till protein. A mutation that has happened in the protein can also affect DNA, but that is a different thing that we're not going to study. But mutation that happens at the DNA level will certainly affect the protein. This is the central dogma. Protein to DNA is not very much possible, but DNA to protein is possible. So that is what they have said over here, that the key differences are not in the alleles of protein coding genes, but in the sequence that decide where and how much the genes are activated. That means there are changes in the sequence of the genes. Okay, now this change can be or can be made in various ways. Okay, so these are the ways in which changes in the sequence of the DNA can be made, and they are given certain names. So if there is a change in the location of the genes, it is known as heterotopy. If there is a change in time, that means with time, if the change has taken place, then it is known as heterochrony. So chrono means what? Time. We see in a chronological order also. What is the chronology? What is the timeline? So heterochrony means change in time. Then amount, if there's a how much amount of the protein ultimately is getting formed, then that is your heterometry. And then change in kind means a different type of protein, if suppose is formed because of the change in the DNA sequence, that is your heterotypy. So these are important terms. Okay, why they are important for this particular part, for this particular chapter? These particular four terminologies are very important, because many a questions will be asked, might be asked from this particular related to these particular four terminologies. So uh, then there is all the examples that has been discussed, that different examples that explains the four different phenomena: heterotopy, heterochrony, heterometry, heterotypy. So these are the four phenomena that has been discussed through several examples over here. Okay, so now what we'll do is that one by one we'll see, when we'll solve the questions, then automatically these examples would become, I mean, the terminologies would become clear to us. Okay, one thing that I wanted to, before starting today's class, I just wanted to give a brief, and also we'll just very quickly see these terminologies as well. Okay, so first is heterochrony. So what is heterochrony? Heterochrony is a shift in the relative timing of two developmental processes. That means, if, just for example, you need not to relate it with the actual embryonic development process. Okay, suppose a particular organ X is has to say is forming in the 10th hour, and the same organ X dash in another body is forming at 12th hour. That means there has been a shift in the timing. That means in animal B, the same organ X dash is taking 2 hours more. That means there is a heterochrony. That means there is a change, there is a shift in the relative timing of two developmental processes. This is your heterochrony. So what is heterochrony? Change in relative time, maybe either in the development of a particular module or in gene expression. So this change can either be in the development of an organism, or the change can also be observed in the expression of a particular gene. So in both the cases, if the change is, if there is a shift in the timing, then it is known as heterochrony. Okay. Oh, sorry. Fine. Then this is your heterochrony. Then heterometry. Heterometry means change in the amount of the protein that has been formed. Okay, I think there is not much detailed explanation here. We'll one by one, well, we'll see. This is important. See here, heterotypy, change in kind. Now when I say change in kind, what does it mean? It means that mutations that actually alter the amino acid sequence of a protein. So if the amino acid sequence of a protein is altered, what will happen? The kind of protein that was formed, that has to form, will not form. The kind of protein would be different. So that is why there is a change in the kind of protein which is formed, because the amino acid sequence of the protein has been altered. Okay, these are the examples. I'm not going in very much detail. So these examples you can cover. I mean, you can read, and I okay. Once somebody's
Simply went that out of water.
um
I must have joining problem solving class.
okay, fine. So now we'll start with the questions. Okay. So the first question is, so as we always do, I'll give the question, I'll give you 2 minutes and maybe 3 minutes for some long questions, and then you can write your answer in the message box. So bats retain interdigital webbing by expressing dash in the wing epithelium. Your options are A, FGF8, B, BMP, C, Wnt, and D, SHH. Okay? 2 minutes to solve this question and then we'll switch the screen. 6:14 right now. I will switch the screen at 6:60. 1 minute more. Okay. Ajay has answered FGF8 and just Reena has also answered FGF8, which is the correct answer. Good. So the question is bats retain interdigital webbing by expressing what in the wing epithelium? So now here FGF8 and BMP, these two options are important because they have kind of antagonist function. How antagonist function? So BMP is basically responsible for triggering the apoptosis of interdigital mesenchyme, which leads to free digit formation. Okay, so if this is your hand, then this is the part. If this skin is there, then it will be like a web. If this is not there, then it will be like a free, it will be like free fingers, free digits are there. So BMP pathway basically it does the apoptosis of these tissues. So are these tissues are removed with the help of BMP pathway. So because it causes the apoptosis of the interdigital tissues. Apoptosis means cell death. Whereas fibroblast growth factor 8, which is FGF8, it is a factor which inhibit apoptosis in the wing epithelium of bats, thus allowing them to retain the interdigital webbing. That means FGF8 kinds of inhibits the action of the uh, BMP pathway. Therefore, BMP pathway is unable to cause the apoptosis of these tissue, and therefore the web-like structure in the case of bats is maintained. Okay. So FGF8 is the correct answer because it plays a crucial role in the development process that leads to formation of bat wings by inhibiting the BMP pathway induced apoptosis. Okay. Now we come to the next question. Which of the following is an example of heterotopy? A. Flipper development in dolphin. B. Limb reduction in whale. C. Loss of vision in cave dwelling Mexican tetras. And D. Development of turtle shell. Okay, it's 6:90. I'll switch the screen after 2 minutes. 21. Hmm. Easy question. And I know that this batch is quite smart and they will be able to answer it quickly, but I'm giving answer time so that everybody can get equal opportunity and equal amount of time to think. Okay. And as this session will also be recorded, so those who are watching through YouTube, they also have ample amount of time to solve the question. One minute more. Okay. Time's up. Let us see what you have answered. Hmm. So Ajay has answered D, that is development of turtle shell, and Jasrina has also answered D, it is the correct answer. Good. So the correct answer is um, hmm, D, development of turtle shell. So it's good that you all are students are reading. So first of all, we should know what is the meaning of heterotropy. So we just explained through the PDF also that it refers to the change in spatial arrangement or change in location of a developmental process. Okay. So when I talk about heterotropy, it is basically change in location. Okay. So now, how development of turtle shell is an example of heterotopy? That means there is some change of location that is taking place because Ribs and other skeleton structure that typically develop internally in other vertebrates have shifted to form the external shells in turtle, representing a change in location of development. Okay, so what does it says? That the skeleton structures in vertebrates, the development of skeleton structures typically happens internally. Okay, it's an internal thing. But in the case of turtle shells, what has happened that this development has shifted uh, from the internal to the external shell, as in external shell in turtles. So therefore it represents a change in location of development. From the internal to external, there is a change in the location of development, and therefore the correct answer is development of turtle shell, and it is an example of heterotopy. Now next question is, what are the adaptations, sorry, what are the adaptations of cave dwelling Mexican tetras? A, properly developed eye, low number of taste buds. B, properly developed eyes, enlarged jaws, increased amount of taste buds. C, absence of developed eyes and enlarged jaws. Oh, sorry. Oh God, you saw the answer. Anyways, D, absence of developed eyes and decreased jaw size. So, I'll give you 1 minute to solve this question because you've already seen the answer. But now that you have seen the answers, which I hope you couldn't, but even though, you also give me at least 2 3 words or one phrase explanation of why you chose whatever answer that you choose. Okay. 6:24, at 6:25 I'll switch the screen. Okay, now I'm switching the screen. I have to be more vigilant while answering. So D, Jasrina has, okay, only Ajay has answered. Good that Ajay has also answered with an explanation. That means that he knows the concept clearly. So he has opted for C, that is absence of developed eyes and enlarged jaws. So what is the correct answer? Yes, it is the correct answer. The correct answer is absence of developed eyes and enlarged jaws. Good. Now let us see what explanation. As these fish are cave dweller, no light there, so don't need eyes, rather the jaw needed for crushing some food. Good. Yes. So it is the correct explanation. So now, what are the adaptations of cave dwelling Mexican tetras? Cave dwelling means uh, basically fishes, I mean organisms that resides in cave. That means that lives in cave. Okay. Now what is Mexican tetra? Mexican tetra is nothing but a freshwater fish. Okay. This is a freshwater fish which is also known as blind cave fish for its inability to see because they don't have eyes. So they are known as blind cave fish. Now always remember that whatever organs that we have in our body, through evolution, through various kinds of adaptations, you must have also heard about the concept of Darwin's finches, that how in relation to the environment that the bird was, the beak of the bird changed uh, to live or to adapt in its surroundings. So similarly, in our body also, we have eyes, we have hands and legs because we need them. There is a need of eye because we have to see things. But can we see very properly at night time? No. So we need to light up, so that we can light up, we need some kind of light so that we can see the surroundings, right? So, but that is just a temporary part. At night we mostly sleep, and then there is day, so there is a day and night cycle. So that therefore the presence of eye is required and justified. But for a fish that lives in a blind, I mean, in a cave, which is completely dark, is there any need of an eye? No, there is no need of eyes, and therefore these fishes, they do not have eyes, uh, but they have a very heightened nervous system and very heightened sensory response. So people who are blind, if you know that people who are blind, who cannot see, they also have very heightened sensory organs. They can very quickly uh, listen to some sounds that we cannot listen, a normal person cannot listen, and they can very quickly respond to other sensory signals. So likewise, in the case of these blind cave fish, that is Mexican tetras, although they do not have eyes, but they have a heightened nervous system, and they can immediately detect where objects or other animals are located by slight changes in surrounding water pressure. Okay, so how they detect that other, or where are the other organisms? By slight changes in the water pressure of the surrounding area. Now, and they have enlarged jaws, just like Ajay. Yes, absence of eye due to high levels of sonic hedgehog. Correct. So that is on the molecular level that you're talking about. So always remember that for the formation of each and every this thing organ in our body, what is needed? Energy is needed. So if and not just for the formation, but for the maintenance of that organ also, energy, a lot of ATP is required. So if a particular organ is not needed, then why to waste energy in first of all making it and then maintaining it? That is why through adaptation, through various molecular pathways, through various molecular mechanisms, it has adapted in such a way that why to waste energy? Instead, we can use that energy in some other way, and therefore, in that way, it has evolved that there are no eyes, but they have enlarged jaws, so that whatever chewing on crushing and predating they have to do, they can do it easily. Then, origin of naked kernel during maize domestication is an example of dash, and it is caused by dash. So your first option is heterochromy and lysine to aspartate conversion, heterotopy lysine to asparagine conversion, heterotopy lysine to asparagine conversion, and heterometry lysine to aspartate conversion. So this should not be a very difficult one, because we have just discussed that what happens when there is a change in the type of change in type, mutation in the type, and it leads to a change in the type of the protein that is produced. There's a change in the amino acid sequence. So I've given all the hints. So it's 6:31. In 2 minutes I'll switch the screen. At least first part should be known to you, that which kind of mutation is happening in this case. Okay. 1 more minute left. Okay, now I'm switching the screen. Hmm. So Jasrina has answered second option, and Aljay has answered heterotopy and lysine to excuse- aspartate. Okay. Okay. So the correct answer is heterotopy lysine to asparagine conversion. Just a minute. Uh, so I think you have tried to answer it correctly only, Ajay. Just just a minute. Hmm. So there are two things, just like a basic uh, concept. There is aspartate and there is. So can you tell me that what is the abbreviated form of asparagine? Hmm? Asparagine is written as ASN and aspartate is written as ASP. Okay? I think you already know this, but I'm just for your confusion per se. So, aspartate is written as ASP and asparagine is written as ASN. And the correct answer is lysine to asparagine conversion. Okay, so it is K to ASN. Okay, I think you wrote aspartate. That is also, I mean, that would create a difference over here. So, just a basic uh, um, re- reminder that you should always try to look these points also. Okay, fine. So now the question is origin of naked kernel during maize domestication is an example of dash and dash. So heterotopy, we have already discussed that heterotopy means changes in the type of changes in the sequence of the protein which lead to altered developmental outcomes. Okay. Now, what happens in maize domestication? First of all, you should know what is the meaning of this domestication. Domestication in general, I think we know what is the meaning of domestication. From wildlife, if you are trying to keep it in a domestic environment, that is a domestication exact the thing. So basically, domestication is a system which can be used for following the evolutionary change. Okay, so what happens that under domestication, wild populations which respond to shifting selective pressures are taken and they are put into a domestic environment, which results in the adaptation of new adaptation to the new ecological niche in which they are. That means that any population, when it is shifted from a wild type to a domesticated kind of an environment, okay, domestic type, then obviously the population has to adapt itself according to the new environment in which it has been put kept. So when it has to adapt itself in the new environment, it leads to new uh, it might lead to new generation of new traits, or it can lead to adaptation which can be at the genetic level also. So because it is so important, the domestication plays such an important role, therefore it has been extensively studied in plant. Okay, And one of the most important or most studied plant is your maize, Z maize, where morphological, genetic, and genomic studies have kind of told or elucidated that how a plant is transformed when it is shifted from wild type to domestic type. Okay, so this is one thing. Now, whenever we talk about domestication, specifically we are talking about the domestication in maize. Okay, so I think you understood what is the meaning of domestication. That means you're shifting a plant from its wild type surrounding to a domestic surrounding. That means you are changing the niche. Niche means the immediate area of that particular plant. So you're changing the niche, you're changing the environment of that plant, and now the plant has to adapt to the new environment. So there would be certain changes that can be tractable. We can track them. Okay, so now when we are talking about the domestication of maize, the most critical step is the liberation of the kernel, liberation of the kernel from the uh, from its case. I mean, from the hardened and protective casing that envelops the kernel. Okay, in the maize progenitor. So this is the most critical step when we talk about the domestication of maize. Now, this evolutionary step which expose the kernel on the surface, this is so important because this leads the maize to be utilized as a food source by humans. Okay, so this key event takes place because of a substitution of lysine to asparagine. Okay, so this hardening of, I mean, the liberation of the kernel from its protective shell, this happens because of a substitution that takes place uh, from lysine to asparagine. Okay, I hope what I'm trying to say it is clear to you. So basically the change that happens, it happens in a gene which is your TGA1 gene. Okay. In this TGA1 gene, there is a lysine to asparagine substitution takes place, which leads to the naked kernel trait, which means loss of the hard protective covering, which is seen in the wild ancestor. The wild ancestor is known as teosinate, teos, teosint, whatever you want to pronounce it as. So important you need to understand is that domestication of any plant in general means taking it out from its wild surrounding and keeping it in a domestic surrounding. And when we talk about the maize domestication, one of the most critical steps of maize domestication is the removal of the maize kernel from its hard protective surrounding, from its hard protective shell, and therefore as a result we get a naked kernel. And this happens because of a lysine to asparagine conversion that happens in the TGA1 gene. Okay, so I think this question is, I hope that this is clear to you. Fine, now we'll come to the next one. Hmm. The cyclodia gene of typical Linaria form the toad of Linaria form of the toad flax carries A. Extensive methylation, B. No acetylation, C. No methylation, and D. Hemimethylation. And it's 6:42. After 2 minutes, I will switch the screen. You have 2 minutes to solve this question. 1 minute more. Okay. Now I'm switching the screen. See what you have answered. Okay. So Ajay has answered C, no methylation, and Jasrina has also answered C, no methylation, which is correct. So can you tell me that what is the effect of transcription in the case of methylation, no methylation, and acetylation in both cases? Can anybody tell me what happens to the transcription of a gene when it is methylated, A, not methylated, and C, acetylated? This is important. That's why I'm asking this question to you. Methylation, no methylation, and acetylation. Can you tell me the difference? What will happen to the transcription? If this is clear to you, then the whole concept is clear of methylation and this thing. Can you think of what should be the correct answer? What should be the possible explanation of it? No? Okay. So, methylation will repress the transcription. Always remember, this is make a thumb rule that methylation will repress the transcription and acetylation will lead to it promotes transcription. So that means if it is not methylated, then what happens? Transcription is getting promoted. Now the question says, the cycloidea gene of typical linearia form of the dot flex carries what? So basically there is no methylation. You have answered it correctly only. It is no methylation, but you should know why no methylation. Because see, सबसे पहले तो you should know what is toad flax. Toad flax is nothing but wild plants, okay, which belongs to the genus of Planaria. And why they are known as toad flax? Because they have flax like leaves and they are basically two-lipped flowers. They have two-lipped flowers. And also, this important to know that these toad flax are invasive species. That means if they are grown outside their native range, then they become very invasive. So this is about toad flax. Toad flax is a wild plants which has yellow, white, or purple flowers. Now, wild type form of typical linearia, it will show dorsoventral asymmetry in both shape and colors of petal. Okay, so there is a asymmetry in the shape and the colors of petals in the wild type form. But in a typical natural occurring mutants, there are certain mutants which are natural occurring, which are more common. They are symmetrical. So why they are symmetrical? This natural occurring mutation, which is the general and the typical form of linearia which is formed, which is present in nature, this natural occurring mutation is not caused by a change in the sequence, but this mutation is a consequence of a epigenetic mutation which involves methylation. So there is an epigenetic mutation that takes place in the natural, and it is a natural occurring, which is the typical form, where there is an epigenetic mutation, and the mutation is in such a way that there is no methylation in the gene. Okay, so in which gene? The cycloidea gene. The cycloidea gene is unmethylated. When it is unmethylated, it will express normally. Okay, so you should always remember that in the wild type there is no significant methylation in the cycloidea gene, and therefore what happens, that this cycloidea gene is actively expressed, and it uh, has to be inhibited, because this cycloidea gene, it controls the symmetry in the flowers. And when in the mutant form, what happens, this cycloidea gene is methylated, sorry, it is not methylated, there is not much methylation. Then what happens, because there is not much methylation, this is expressed normally, and therefore typical bilateral symmetry is there. When it is extensively methylated, it will be silenced, and when it is silenced, then symmetry is not maintained. So to maintain the symmetry in the normal form, the gene should not be methylated, so that the gene can be expressed and symmetry is maintained. But in the mutant form, what happens, that this gene is mutated, because this gene gets methylated, and in the mutant form, see, in the here it is saying typical form, not the mutant form. In the mutant form, when the gene becomes, cycloidea gene becomes heavily methylated, then the symmetry is not there. Okay, next question is, which of the following statements regarding evolutionary changes are true? A. Inhibition of apoptosis by BMP is required for bat wing development. B. Ectopic expression of FGF10 in the tongue would result in the formation of calcareous structures. C. Pythons possess a rudimentary femur due to posterior HOXC8 and HOXC6 extension. D. Overexpression of FGF8 in humans would result in longer digits. And E. In kangaroos, heterochrony of the forelimb is required for them to crawl in the pouch. So I'll give you 3 minutes to solve this question at 6:50. I'll switch the screen at 6:53. Okay. Now try to solve this question. Okay, now I'm switching the screen. Let us see. Okay. Okay. Why this question is in evolution? Uh, which question? This methylation question you're talking about. So, in a way, they are kind of related to evolution because it shows that how the various organs and how the various features have developed and what are the reasons for their development through time. So, it focuses on the fact that not only the gene the gene sequence is important, but also the epigenetic changes that happens during evolution cannot be neglected. So, therefore, one must also acknowledge the fact that it's not only the genetic expression changes, the sequence that is important, but the epigenetic changes which is very important. Now epigenetics is a very evolving field. If you see, epigenetics is the new the new boost, we can say, because if we want to study the expression of a particular gene in totality, we not only need to consider the sequence of the DNA, but also the whatever forces or whatever interactions it has with the environment as well. Therefore, it is epigenetics. It is even more important. Anyways, so now, so Ajay has answered B. Okay, okay, you have answered that B and D are the true statements. Hmm. B is ectopic expression of FGF10 in the tongue would result in the formation of calcareous structures, and you have answered D and E also. Okay, okay, E also. D is overexpression of FGF8 in humans would result in longer digit, and E, that is in kangaroo, heterochrony of the forelimb is required for them to crawl in the pouch. So let us see what are the correct answer. Very well, the correct answers are this only. Ectopic expression of FGF10 in the tongue would result in the formation of calcareous structures. Overexpression of FGF8 in humans would result in longer digits. And in kangaroo, heterochrony of the forelimb is required for them to crawl in the pouch. Fine. Now let us try to understand the question, and we'll try to understand in brief each and every option. Okay, so first option is inhibition of apoptosis by BMP is required for bat wing development. Now we have just studied that if we want webbed structure or a wing structure, do uh, what do we need? Do we need BMP? No, because BMP actually promotes apoptosis. Here it is written that inhibition of apoptosis by BMP. No, BMP is not inhibiting apoptosis. BMP actually supports apoptosis. So BMP actually promotes apoptosis in the interdigital regions of most of the vertebrates. For example, we studied in bats also that if we want a webbed structure, then BMP signaling has to be inhibited, because if the BMP signaling is inhibited, then only apoptosis can be inhibited, and then only your webbed structure could be formed. See, here we discussed about this. So this means that first option would be false. It is wrong. Then next is ectopic expression of FGF10 in the tongue would result in the formation of calcareous structures. So it is correct because FGF10 is involved in organogenesis and limb development, first of all. It can potentially induce the formation of bone like or mineralized structures in the soft tissue, such as tongue. Okay, so Therefore, if there is an ectopic expression means ectopically we are expressing from the outside, extrinsically we are giving FGF8, FGF10. Okay, so we are providing FGF10 from outside. We are extrinsically providing FGF10 in the outside in the tongue region. So since FGF10 is involved in limb development, in organogenesis, therefore it can potentially induce the formation of bone like structures or mineralized structures like calcareous structures in the tongue. Also, FGF10 it plays a very important role in mesenchymal to epithelial signaling during organ development. Therefore, if FGF10 is misexpressed, it can also lead to abnormal tissue formations. And this abnormal tissue formation can also resemble calcified elements. So, just a minute, I have a call. Hello, sir. Hello. Yes, sir. Yes, sir. That point is that we have to discuss. Uh, yes, sir. Tomorrow, first thing we'll discuss this only, sir. I'll also see. Yes, sir. Yes, sir. I'll remind you for sure. First thing in the morning. Okay, 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 sir. Okay, sir. Yes, sir. Okay, sir. Okay, sir. I'll check it. Okay, sir. So sorry for the disturbance. Actually, my PhD supervisor called me for some work. So, anyways, so where were we? We were discussing the option B. So yes. So yeah. So we discussed, I think, the B, that B is the correct answer. B is the correct option. Then C. What is C? Pythons possesses a rudimentary femur due to posterior Hox C8 and Hox C6 extension. So basically, Hox genes are involved in vertebrate body plan development, that we know, but it is not specifically the posterior extension of Hox C8 and Hox C6 that results in python rudimentary limbs. Okay, so exact mechanism is more complex and it might involve other Hox genes and it might also involve other Hox factors also. So therefore, it cannot be it's not the only thing that is important, I mean, that is needed for the rudimentary femur in the case of pythons. So you just need to understand that the mechanism for limb reduction in pythons, it involves more complex genetic changes beyond hox6 how in hox c6 and hox c8 expression. So this is not right. Then overexpression of FGF8 in humans will result in longer digits. Yes. Why? Because what is the function of FGF8? So this FGF8 is involved in limb bud formation and digit growth during the embryonic development. It is involved in the growth of the digits. Okay. This FGF8 is responsible for the growth of the digits. So if there is an overexpression, obviously it will result in longer digits. It's easy. Then in kangaroos, heterochrony is of the forelimbs is required for them to crawl down um, into the pouch. Yes, it is also true. So basically, what is heterochrony? Crony. See, if there are so many words like this and you get confused, okay, by heterotopy is also there, heterometry is also there, heterochrony is also there. What is what? So you can have certain tips and tricks by which you can remember it. First is heterochrony. It's very easy. Why? Because I already told Chrony means chrono. Chrono means order, means time, the chronological order, the timeline. So from that you can remember heterochrony is something which is related with time. So Heterochrony is basically the changes in the timing of the development process. When there is a shift in the timing of the development process, then that is known as heterochrony. Now the option says that in kangaroo, heterochrony of the forelimb is required for them to crawl into the pouch. That means there has to be a change in the timing of the development of forelimbs. So is it correct? Yes, because the forelimbs develop early and are functional at birth, which allows the newborn to crawl into the mother's pouch. So as soon as the baby is born, the kangaroo is born, the forelimbs are already developed, and because they are developed, that is why they can crawl into the pouch. So there is a shift in the development of the forelimbs. They are developed early, which helps the kangaroo to go into inside the pouch, mother's pouch. So this is an example of heterochrony. Okay, I hope this is clear to you. Hmm. Okay, next question is, which of the following statements about mechanisms of evolutionary changes are true? A. Cyclopia is a result of the absence of heterometry. B. Methylation of coding sequences is a conserved phenomena for ensuring tissue specific expression in vertebrates. C. SHH expression is higher in human digits as compared to whales. D. An increase in the jaw size in cave fish is a result of PAX6 heterotopy. And E. Heterotopy of cis-regulatory regions are responsible for the wide array variety of limbs in orthopods. This is the question. You have 3 minutes to solve this question. This is right now it is 7:05. I will switch the screen at 7:08. Okay? Try to solve this question. 2 minutes more. 1 minute more. Okay, your time is up. I'm switching the screen. Okay, fine. Let us see what you have answered for this question. These MCQ are very difficult. Not difficult for you to answer, but for me to explain, I have to speak so much continuously for 2 hours. Okay. So, okay, let me see. 7 4. Physical constraint. Hmm. Cyclopia is a result of essence heterometry. Okay, A, you are saying. Okay. B, methylation of coding sequences is conserved. Okay. And Jasrina has answered A and E. Fine. So let us see the correct options are cyclopia is a result of absence of heterometry, and B, that is methylation of B, it is B, no? Yeah. B, it is methylation of coding sequences is a conserved phenomena for ensuring tissue specific expression in vertebrates. Okay. So now let us try to understand this question. So we have to pick the correct statements regarding the mechanisms of evolutionary changes. So first is cyclopia is a result of absence of heterometry. So what is cyclopia? Cyclopia is basically associated with disruption in the development of forebrain. Okay, particularly the failure of proper patterning, which can be described as change in the size or proportion of structure. Therefore, we call as heterometry. Therefore, the absence or disruption in the normal measurement of development, it can lead to conditions like cyclopia. Understood? So I think you already know because you both have answered it correctly. So the first is correct. Okay, so it is basically disruption in the development of the forebrain. There is a failure of proper patterning, and therefore it can be uh, there is a um, change in the proportion or the size of the structures, which is a concept which is related to heterometry. So let us see this. Just 1 minute. Not this. Not this one. Yeah. So here you can see heterometry means different part grow at different rate. There's a change in the amount. So that means the size and the proportion of structures are different. So in cyclopia, what happens? That because there is a failure in proper patterning, therefore there is a change in the proportion or size in the development of the forebrain. So therefore first is a this. Then methylation of coding sequences is a conserved phenomena for ensuring tissue specific expression in vertebrates. Yes. So methylation play a significant role in gene regulation, and this is indeed conserved among vertebrates and which contribute to tissue specific expression patterns. So we all know that the genes are expressed uh, or rather genes are differentially expressed related to their cell type and their tissue location. So this is differential expression of the same gene in different location is governed by various factors, and one of the factors that governs the differential expression of genes is the epigenetic marks that are present. So if a gene is heavily methylated, that means the gene is not going to be expressed in that particular tissue. And if the gene is not methylated, that means the gene is going to be expressed in that particular tissue. Or if the gene is acetylated, that means it is going to express. Now, when I talk about the expression, you should also consider in mind that this methylation patterns can be present two ways at two places. Can you tell me? This is a very basic question that I'm going to ask, yet a very interesting one. That if I say that methylation of the sequences, so where do you think what kind of methylations I am talking about. Methylation of coding sequence. That means where you can think in the sequence methylation can happen. Hmm? Where you think in the sequence actually uh, physical constraint for kangaroo, I wanted to know. Ajay, please elaborate your question in the message box, okay? Meanwhile, also try to answer this question that I have just put. That when I say that methylation of coding sequence is happening, methylation of the sequence is happening, then where methylation is happening? In the sequence, what is getting methylated? Which part of the DNA sequence is actually getting methylated or demethylated? In normal ATGC, okay, which is which gets methylated the most, that you have studied also, you must have studied. Hmm? Which part gets methylated? Don't know? Yes, histone and cytosine. So when we talk about the DNA sequence, it is basically the cytosine that we're talking about, because histones would be proteins. Okay, these are proteins. So we're talking about DNA. So in DNA specifically, we talk about cytosines. So that means the CPG islands are getting methylated. That means whatever methylation marks in the DNA are present, they are present at the CPG islands in the DNA sequence. In the DNA sequence, there are certain C and G rich repeats or CG rich sequences, which are known as CPG islands, where your methylation happens. In DNA, when we talk about proteins, then we talk about mostly histones methylation and all. So that this is just, I wanted to clear that methylation can happen on histones also, methylation can happen on DNA also. If methylation happens in histones, that means where in the histone, basically the tail of the histone gets methylated. So histones ka tail is the one which gets methylated. For example, if I write H3K4Me3, that means histone number 3. Okay, just a minute. Hmm. That means histone number 3 and lysine, fourth lysine. That means fourth lysine of the histone 3 has 3 methylation, is trimethylated. So this is how we keep methylations on histones. But here when we're talking about of in DNA sequences, methylation happens at the C cytosine residue, basically in the CPG islands. Okay, so the B option is correct. Then third is SHH expression is higher in human digit as compared to whales. No, this is false, because because if we see whales, then they have gone extensive modifications that leads to the reduction in the limb structures. Okay, so this becomes your wrong answer. I'm sorry, I'm unable to speak because I'm not feeling that well and you have to speak so much. Yeah, fine. Correct, Ajay. Then, an increase in jaw size in cave fish is a result of PAX6 heterotopy. So it is also a false statement. Why? Because as we have mentioned in there was earlier question also, that jaw size of cave fish is more, and this change in the jaw size of cave fish they are actually influenced more by Hox genes rather than PAX6. So PAX6 is primarily involved in eye development. It is development of the eye, whereas Hox genes are more responsible for the increase in the jaw size. So this is becomes false. Then heterotypy of cis-regulatory regions are responsible for the wide variety of limbs in orthopods. It is also false. Why? See, cis-regulatory elements, they contribute to limb diversity, that is okay. But this statement is not complete because here the relationship is not mentioned. It is an incomplete statement according to me, because what kind of relationship they wanted to tell, that is not mentioned here clearly. So that is why this is not a very correct statement. Okay, fine. Next question. Okay, so Ajay, this physical constraint for kangaroo, I couldn't understand your question. So please elaborate your question in the message box so that I can try to answer it. Okay, what you wanted to ask, I couldn't understand. So this the option about this kangaroo thing, it was just that when kangaroo is born, the forelimbs they develop. There is no kind of constraint there. There is no kind of constraint in kangaroo. You just have to remember that uh, the phenomena which is called as heterochrony, it is observed in the case of kangaroos. Okay, so what does heterochrony refer to? It refers to change in the timing of developmental process. That means at birth the four limbs of the kangaroo, they are developed and they are functional. Okay, so at the timing of birth also, in compared to the other parts of the kangaroo's body, the four limbs are more developed and they are functional even at the time of birth. And why so? Because with the help of four limbs only, kangaroo jump and go inside the I mean, not jump exactly, but they crawl and go inside the mother's pouch. So that is the reason why the and in the pouch they reside, in the mother's pouch, baby kangaroos reside. So that means with it, in respect with all the other body parts, with respect to all other body parts, the four limbs are more fine developed even at the time of birth, and therefore there is a change in the timing of the developmental process. That means four limbs are developed early and other parts of the body eventually develops. So because there is a change in the timing of development process, therefore this is known as heterochrony. This phenomena is heterochrony, because there is a change, there's a difference in the timing of the development of one organ to another organ, or one body part to another body part. So this is your heterochrony. There is no physical constraint as such in kangaroos. Okay, and according to this question. Now next question is, given below are the beaks of finches mutated during embryogenesis. Based on your understanding of heterometry, identify the false statements. Okay, false statements you have to identify. Note it. First is, bird A was incubated with calcium sequestering molecules. Then, bird A may have had an overexpression of BMP4. C. Bird A may have had an overexpression of calmodulin. D. Bird B may have had a gain of function mutation in the kinase domain of calmodulin. And E. Bird B may have had a gain of function mutation in BMP4. So, this is your question. Right now it is 7:23. I'm giving you 3 minutes. So I'm going to switch the screen at 7:26. Read the question carefully. See the image that is given to you. If you have the basic concept of BMP4 and calmodulin, which has already been taught in the PDFs, I mean, in the recorded lectures, then it would be easier for you to answer this question. Okay, so try to solve it. 3 minutes you have. 2 more minutes. Okay, your time is up. Okay, let us see. Hmm. Okay. Ajay has answered except C. I think all are false. Justina has answered A, B, and D. Okay. The correct answer are this. Bird A was incubated with the calcium sequestering molecules. Bird A may have had an overexpression of BMP4. Bird B may have had a gain of function mutation in the kinase domain of calmodulin. Bird B may have had a gain of function mutation in BMP4. Okay, these are the correct answers. So now let us try to solve this question. So, yes, let me just make it a little bit clear for you. I'm sorry, I'm not very good in drawing, especially when I'm using this pen for the PPT, but I'll try. If this is your finch beak, okay, so this particular is the length. This is the depth, and this represents the width. Okay, this has already been taught to you in the recorded lecture. Now there are two things. First is BMP4. Okay. And second one is calmodulin. Fine. This BMP4 basically promotes beak width and depth. So what does it do? It promotes beak width and the depth and the width. That means if there is low BMP, then what will happen? Low BMP4, then there would be low depth and low width. Then your calmodulin, this calmodulin influences beak length. This calmodulin, it influences beak length. So what does it mean? That if there is low calmodulin, that means it will have a short beak. Low calmodulin, if calmodulin is low, that means it has a short beak. Calmodulin is what? It is, what is calmodulin? Basically, calmodulin is calcium attaching or associating molecule that binds to calcium. Okay, now we'll see the options. By one, we'll try to see the options. First is bird A. So now you can see here that with respect to control, the width of the bird B is very less. That means it can have less of BMP or not. Let us say, I don't want to confuse like this. Okay, so the first option is bird A was incubated with calcium sequestering molecules. So is this true or false? It is false. Why? See, calcium sequestering molecules would reduce the calmodulin effect. Why it will reduce the calmodulin effect? Because calmodulin actually binds to calcium and then it does its function. So if there are other molecules that have already sequestered or already taken up the calcium, then calmodulin cannot function properly. So if calmodulin is not function properly, that means what will happen? Calmodulin is required for the beak length. Here you can see that the length of this A is very much greater as compared to control. And calmodulin is required. So that means if calmodulin is less, then can this beak be of this much length? No. That means excuse me, that means that calmodulin is functional over here. And if calmodulin is function, that means calmodulin is able to bind to calcium. And if it is able to bind calcium, that means calcium sequestering molecules are not there. So this becomes false. Okay, because if there are calcium sequestering molecules, then how calmodulin will become functional? So this is your false statement. Then bird A may have had an overexpression of BMP4. This is again a false statement. Why? Because the, see, this is the depth and this is the width. Okay. So overexpression of BMP4, what will happen? The beak should be more wider and more deeper. But can you see it as a more deeper width? No, it is not deeper. Uh, yeah. But can you see it as wider and deeper? No, it is neither wide nor deeper. So this bird A has an elongated peak. Since it has an elongated peak, how can we say that there is an overexpression of BMP4? Had it been that BMP4 has overexpressed, then it should be wider than the control and deeper than the control. So this is again false. Okay. Bird A may have had an overexpression of calmodulin. It is true because calmodulin is responsible for increasing the beak length. More the calmodulin, more would be the greater would be the beak length. And as you can see that the length of the beak is more in respect to control. So this can be the true statement. Okay. Then bird B may have had a gain of function mutation in the kinase domain of cadmodulin. So if the kinase domain of the cadmodulin is there is a gain of function mutation, that means the effect of calmodulin should be even more. And if the effect of calmodulin is even more, then the length of the beak should be very large because calmodulin is required for beak length. But here you can see that the length of the beak has even reduced with respect to control. So that means this statement would be false. Then bird B may have had a gain of function mutation in BMP4. So if there is a gain of function mutation in BMP4, so BMP4 gain of function mutation affects beak ka width and beak depth, which does not align with the bees bird's wider beak. There is no depth here. So that's why it also becomes false. So that means only C is correct, rest all are false. Okay, fine. Now we come to the next question. Given below are some statements regarding mechanisms of evolutionary changes. First, sickle cell anemia is caused by heterotopy of beta beta hemoglobin. B. Hemophilia A is a result of heterometry of factor 8. C. Third, most cancers are caused due to heterometry of P53. And fourth, thanatophoric dysplasia is caused by heterochrony of FGFR3. Choose the option with all the correct statements. Now you have to see that what all are the correct statements in these many options. So these many options are given. I think all the possible options they have tried to given. Anyways, so you have 3 minutes to solve the question. Not to solve the question, but to select the correct options because there are so many of them. So right now it's 7:36. I'll switch the screen at 7:39. Okay. Try to solve. 2 more minutes left. 1 more minute left. Okay. Okay, now I'm switching the screen. Hmm. Let us see what you have answered. So just Reena has answered all correct, and Ajay has answered one, one, three, and four. Okay, one, three, and four. Yeah, okay, fine. I think it's right. One, three, and four. I think is the right answer. Yes. So the correct answer is one, three, and four. These are the statements which are correct. One, three, and four only. This one is the correct statement. And Jasreena has answered what? Jasreena has answered as all correct. Okay, now we'll see the explanation. I think Jasreena, then you will get to know the correct thing. And Ajay has answered hemophilia is classical absence of factor 8. Hmm. Okay, good. Good that both are participating today. Okay, so the first is sickle cell anemia is caused by heterotypy of beta beta hemoglobin. I think you all know it is correct because cell anemia is caused by a mutation in the beta globin gene that we know, and it leads to the development of abnormal hemoglobin, which we call as, I mean, write as HbS. So this is an example of heterotypy. Why? Because there is a change in the type of the protein produced. Okay, because of the mutation, the type of the protein that is produced is changed. So this is your true. This is true. Then hemophilia A is a result of heterometry of factor B. It is also correct. Sorry, it is false because hemophilia is caused by mutation in factor 8 gene, which leads to deficiency of this clotting factor. We know this. So this is not heterometry. Okay, heterometry is, it refers to change in the amount in the level amount of particular protein or expression of a particular level of expression of a particular protein. But here it is just that there is a mutation in the factor 8 where there is a deficiency of clotting factor. So we cannot relate it with heterometry. Okay, because we don't know that the amount or the level of the expression of the protein, what happens to that. Then most cancers are caused due to heterometry of p53. So we can say, because heterometry is what it is basically, change in the level of expression or level of or the amount, that is your heterometry. Now there is, in the case of cancer, what happens is there is alteration in the p53 expression, which can also lead to their loss of function. Therefore, this can be associated with heterometry because there is alteration in the level of expression of p53 in the case of cancer. So this is can be associated with heterometry. Then your fourth is thanatophoric dysplasia is caused by heterochrony of FGFR3. So this is also true. Why? Basically, when there is a mutation in the FGFR3 gene, what happens? It disrupts the normal bone growth, and when the normal growth of the bones is disrupted, then there are changes in the developmental timing. Okay, there are changes in the developmental timing, and when there are changes in the developmental timing, then this that phenomena is known as what? That phenomena is known as heterochrony. Therefore, fourth option is also correct, that thanatophoric dysplasia is caused by heterochrony of FGFR3 because Due to the mutation in FGFR3, the normal growth of the bone is disrupted, which can change the developmental timing. So it is correct. So the correct answer is 1, 3, and 4. Okay, fine. I think this is the last question for the day. Yes, chrony means time, heterochrony means time. Correct. Okay, God, these kinds of questions where you have so many options, these are very tiring and cumbersome questions. Anyways, given below are some statements on developmental constraints on evolution. Choose the correct statement. See, developmental constraints on evolution, and you have to choose the correct statements. A. Turing's model can explain the striped pattern of a zebrafish. B. A loss of function mutation in phenylalanine hydroxylase would have pleiotropic effects. C. A loss of function mutation in DNMTs would inhibit epiallelic phenotypes. D. Susceptibility to ulcerative colitis and Crohn's disease due to differences in gut microflora is a result of symbiont variation. And finally, the last statement E. Industrial melanism, which caused an increase in the population of dark peppered moths, was due to environmentally induced genetic variation. Okay, just a minute. Um, is this happening or not? Yeah. So this is your question. I'll give you 3 to 4 minutes to solve this question because so see, these questions are not difficult but time consuming because like I said in the last class also, that generally in multiple choice questions you can have like 3 4 options. Mostly 4 options are only there, and in the majority cases those 4 options are somewhat the way related to each other. Like maybe those 4 options are related to any one pathway or any one particular phenomena. But here, all the options, first of all, they're not just 4 options, you have more number of options. I think in all of the questions, in most of the questions, you have more than 4 number of options. And then all the options are different concepts. You see, mostly in MCQ, you can have different options from the same concept. But here you have different options and all are different concepts. So if you are able to solve one question, that means you are thorough with one, two, three, four, five concepts. So just solving these questions will uh, uh, make you strong in not just one but in 4 topics. So that's why these questions are important and a little tedious also for me to explain as well. But anyways, you can I'm giving you 3 to 4 minutes to solve this question. Just go through all the options very carefully and then try to answer it. Okay. Chalo, the time starts now. It's 7:48 right now. I'll switch the screen in 3 minutes. minutes more. 30 seconds more. Okay, now I'm switching the screen. See what have you. Hmm. Okay. A reaction diffusion model, correct. Jasrina has answered A and D. Ajay has answered B. A, B, C is correct. Okay. So Jasrina has answered A and D. And Ajay has answered A, B, and C. Fine. Let us see what are the correct options. So these are the correct options. During's model can explain the striped pattern of a zebrafish. This is correct. A loss of function mutation in the phenylalanine hydroxylase would have pleiotropic effects. This is correct. A loss of function mutation in DNMTs would inhibit P-allelic phenotypes. This is correct. So you can tally whether your uh, answers are correct or not. Okay. So let us try to understand this question. Last question for the day. Hmm. First is Turing's model can explain the stripped pattern of a zebrafish. So is it correct statement? Yes, it is a correct statement. This is so basically what is Turing's model of morphogenesis? Turing's model of morphogenesis basically describes how chemical substances, that is morphogens, sorry, how chemical substances such as morphogens can interact to create patterns in biological substances. Okay, so Turing's model of morphogenesis, there is a model which is known as Turing's model of morphogenesis. It describes how chemical substances can interact with each other to create biological to create patterns in biological systems. Now, It has been applied to explain the striped pigmentation pattern which is seen in zebrafish as well, which is nothing but a result of interactions between the pigment producing cells. Okay, so that means when morphogens interact with each others, they create a striped patterns in biological system. So this is explained with the help of a model which is known as Turing's model, which is a result of interaction between the pigment producing cells. Then next is B, a loss of function mutation in phenylalanine hydroxylase would have pleiotropic effect. So this is true or not? It is true. So why? Because a loss of function mutation in phenylalanine can lead to phenylketonuria. Okay? And which can have various effects on metabolism, brain development, and overall health. So, first of all, you should know what is the meaning of pleiotropic effect. Pleiotropy means one gene can affect multiple traits. That means one gene is able to affect multiple traits, then that effect is known as a pleiotropic effect. So, when there is a loss of function mutation in phenylalanine hydroxylase, it leads to phenylketonuria, which can have effects on metabolism also. It can have effects on brain development also. It can have effect on the overall health of the organism also. That means there is one gene that has been affected, that is phenylalanine hydroxylase, but the effect of one gene is affecting metabolism, brain development, and so many things. Therefore it is an example of pleiotropic effects. This is correct. Then a loss of function mutation in DNMTs. What are DNMTs? DNA methyl transferases. So a loss of function mutation in DNMTs would inhibit epiallelic phenotypes. So what is the meaning of epiallelic phenotypes? That means epiallelic phenotypes. That means phenotypic variations due to epigenetic changes. Okay, what is the meaning of epiallelic phenotypes? I'm again repeating it. Listen very carefully. Phenotypic variations due to epigenetic changes. So the option says a loss of function mutation in DNMT, that means DNA methyl transferase, will inhibit the epiallelic phenotypes. It is true or false? It is correct, because DNA methyl transferase will add what does they do? So DNA methyl transferase, that means these enzymes are responsible for transferring the methyl groups to the DNA. That means they add the methyl groups. So when DNMTs add methyl groups, they will have some epigenetic modifications. Correct? So if there is a loss of function of DNMTs, that means methylation is not going to happen. That means the epigenetic modifications are altered. And when the epigenetic modifications are altered, that means the epiallelic phenotypes, that means the variations due to epigenetic changes, are also inhibited. So, a loss of function mutation in DNMTs would indeed inhibit the maintenance of these epiallelic phenotypes. Therefore, this is a correct option. So, I hope this is clear. Then, next is susceptibility to ulcerative colitis and Crohn's disease due to differences in gut microflora is a result of symbiotin variation. It is true or false? It is false. Why? Because gut microbiota plays a very important role in susceptibility. It's fine, but the statement may imply that this is purely due to symbiotic variation without considering the complex interplay of other factors. Okay, and what are those other factors? Those other factors are genetics, your immune response, your environmental factors. So yes, you can say that gut microflora plays a very important role in making an organism susceptible to ulcerative colitis and Crohn's diseases. But the susceptibility to ulcerative colitis and Crohn's diseases is not only due to difference in the gut microflora. But there are other factors also which contributes to it. And what are those other factors? Those other factors are genetics, responses, and environmental factors. So there is a complex interplay between these factors that leads to this ulcerative colitis and Crohn's disease susceptibility. Yes, Ajay, you can, whatever is your query, you can please type it in the message box.
Actually, gut microflora makes resistance for this ulcerative colitis and Crohn's disease. It,
Yes.
gut microflora don't make
See,
susceptibility, which it don't increases the susceptibility, it increases the resistance because of result of symbiotic variation.
huh, but you
Symbiosis
have
is
not.
there, gut flora, but the
Okay,
susceptibility word is
Aj-
wrong.
Ajay, listen to my what I am trying to say. That here it is written differences in gut microflora. That means the normal gut microflora, which is good, which is helpful in making the your gut resistant to ulcerative colitis and Crohn's diseases. Okay, but if the normal gut microflora, which is indeed important and necessary for resisting the gut from ulcerative colitis and Crohn's disease, is altered, if there are differences in gut microflora, see, you have to read the question very carefully. Due to differences in gut microflora, so in normal condition, gut microflora is responsible for maintaining a proper and a healthy gut. That is why there is a gut microflora. But due to certain, for example, if if you are eating very spicy food, or there are so many reasons, if your gut microflora is disturbed, if your normal gut microflora is disturbed, there is a difference in the normal gut microflora, then you will become more susceptible to ulcerative colitis and Crohn's disease. This is what the question is saying. Okay, the option is saying, okay, it is not saying that normal gut microflora will make it susceptible. It is saying that if there is a change, for example, if there is a gene which is very much important, and there is a mutation in that gene, that mutation in the gene can lead to a diseased condition. Similarly, just like a mutant condition, like a mutation condition, if the normal gut microflora, which is indeed responsible and very helpful for maintaining a proper healthy gut, is disturbed, then the difference in the gut microflora will make you susceptible to ulcerative colitis and Crohn's diseases. Okay, so you see, read the sentence. That's what I say, that your concept can be completely right, but you have to see what they are asking in this question. What is the context of the question? What Whatever information you have, that you have to apply and see how the question is framed. They have framed the question very smartly. See, susceptibility to ulcerative colitis and Crohn's disease due to differences in gut microflora. Okay, they have not written that due to normal microflora. Okay, now last one is industrial melanism, which caused an increase in the population of dark peppered moths, was due to environmentally induced genetic variation. So this is again a false statement. Why this is false? Because industrial melanism is primarily explained by natural selection, which acts on the existing genetic variation rather than the environmentally induced genetic variations. So natural selection works on your existing genetic variation, not on the environmentally induced genetic variation. So therefore, this statement also becomes false. So with this, we have completed today's lecture. Last lecture would be, the next week would be a last lecture where we would kind of try to phenotypic. Yes, phenotypic, you are correct. Phenotypic changes are not there. So in the next week's class, we'll just try to revise. It will be like a summary class for the next week. So thank you so much, everybody. We'll meet in the next week.